Good morning, all. Molecular markers. Today topic important one. Molecular markers. So first of all, let's know about the definition of molecular markers. Actually, the molecular markers are is a short fragment of DNA that associated with certain location within the genome. These molecular markers are used in molecular biology, biotechnology to identify the particular sequence of DNA in pool of unknown DNA. That is the benefit importance of this DNA molecular DNA. So simple definition: these molecular markers is a gene or DNA sequence with a known location on chromosome that can be used to identify cells and individual species. So it is just a piece of DNA. It is a just piece of DNA that is called DNA molecular markers. So what are the ideal markers? The ideal markers should be highly polymorphic nature, codominant inheritance, frequent occurrence in genome, easy access, easy to fast assay, high, high reproducibility, easy exchange of between laboratories, gene-based molecular markers. These are ideal DNA markers characters. So first of all, highly polymorphic. Highly, where they give it, just wait. Uh, Constraints, morphological markers, types, properties, ideal, right? Right. So what is it highly polymorphic? It must be polymorphic as it polymorphism that is measured by genetic diversity study. What is polymorphism here? Polymorphism is a detectable and heritable variation at a locus. That is polymorphism. So this is mainly useful for measured for genetic diversity studies. Codominant inheritance. Codominant inheritance determine of homozygous and heterozygous states of diploid organisms. This is for this nature for determination of homozygous and heterozygous state of uh, diploid organisms. Frequent occurrence in genome, a marker should be evenly and frequently distributed throughout the genome. That is also another character. It should be in easy access, easily availability. It should be easy and fast and cheap and detect. Easy and fast assay. High reproducibility. And it consists high reproducibility. That means it should give similar result in different experiment irrespective of time and place. That is reproducibility. Easy exchange data between laboratory and gene-based molecular marker. It should be gene-based. Already told you in the definition itself, the short genes, short fragments. Non-PCR hybridization based markers. Uh, Actually, before that, the markers, let's see some different type of markers here. Okay. But in our book phenotype, the protein, morphological protein, different, no need that one. Types of DNA molecules. Actually, these are two types: coding, dominance, and codominance. There, dominant, codominant. First of all, AFLP, dominant, dominant type. AFLP and RAPD, ISSR, SSR. Sir, not SSR, ISR. So these are dominant. Codominant types are RFLP, RFLP, short nucleotide SNP, SSR. These are codominants type of gene molecular. And another type here, the gene moleculars are hybridization based molecular markers. Hybridization, one is hybridization based molecular markers. Second one, PCR based molecular markers. First, hybridization molecular markers, only one example, that is RFLP. RFLP hybridization method. RFLP restriction fragment length polymorphism. I will explain later. And most of the remaining PCR based molecular markers. RAPD, RAPD, random amplified polymorphic DNA, and these are most of the, for example, this one of the example for PSR based molecular markers. Here, molecular markers, no need to explain all, just only two or three important molecular markers I will explain, right? So, non PCR or hybridization. Hybridization, non PCR, both are same. Now, let's see non PCR hybridization based molecular markers. Here, just wait. So the variation in length of DNA fragment produced by specific restriction endonuclease from genomic DNAs of two or more individual species is called hybridization. And marker produced by the technique are called hybridization based molecular technique. So that means the variation in the length of the DNA fragment produced by specific restriction enzyme, the endonuclease. For example, if we use restriction endonuclease from the genomic DNA of two or more individuals, here same restriction endonuclease, here one individual, second individual, here it has the some DNA molecules, it has also DNA molecules, but you use digestive enzyme restriction endonuclease enzyme for the both the same enzyme adding. Here also same enzyme added to this from DNA two or more individual species called hybridization. That means when you use this restriction endonuclease to different individuals, after, with help of this restriction endonuclease, short fragments are formed. Short fragments are formed. Here also from short fragment. These short fragments called as a markers. These short fragments called as markers produced by this technique are called hybridization based molecular techniques. So this techniques called as hybridization based molecular techniques, right? That means actually in these two different individuals utilized. That's why this hybridization method. In this method, The best one of the best example in this non-PCR hybridization method, RFLP. This is very important for long stay. RFLP genetic markers. RFLP actually it is a piece of DNA that cut from larger DNA molecule by restriction enzyme. The type polymerism that result from variation in the DNA sequence recognized by restriction enzymes. So the genetic marker resulting from variation here, genetic marker resulting from variation or change in the length of defined DNA fragments produced by digestion the DNA sample. When you utilize the restriction endonuclease, it gives the fragments, but in this fragments shows the variations and changes in length, variation and changes in length. That's why it is called as a polymorphism. Here given, for example, in diagram given clearly A, B, C, D, E, G. Now, these are the locations. After utilize the restriction endonuclease, actually these are the red color restriction site. Yeah, on the restriction site, with help of restriction endonuclease enzyme cuts into fragments. If you see the fragments, is there all same? No. A small in size, B somewhat large, C1 large, and D medium, and E some more large, and G even large, and F. So these like different, that not unique fragments are produced. Different shape of different length of fragments are produced. That's why it is polymorphic. This restriction fragment length polymorphism, electrophoretic comparison is the size defined. Uh, the first step in this restriction endonuclease, in first step, the isolate high quality DNA, that is the first one. From the organism, from the cell, we should separate high quality DNA. Second step, digest with combination of restriction enzymes. Combination, not unique. Combination, that hybridization means combination. So digestion of the combination of restriction enzymes. Next, fractionate digested sample by electrophoresis. I will explain later electrophoresis. Transfer fragments to membrane. Hybridize radioactively labeled DNA probe. Detected by autoradiography. Can also use non-radioactive labeling system also. So apart from that, in RFLP, the clear, mostly actually in our book, given clearly in RFLP, what they given. First of all, steps. The following basic step in RFLP, that is DNA isolation. First, as they given, no need to all this one. First, okay, this is right. DNA isolation, isolate DNA. Cutting DNA into smaller fragment using restriction enzyme. Second, digest, that is nothing but cutting with help of the different combination of restriction enzyme. After cutting, actually separate. Fractionate means separate. Here separate, the best word, separate. Separate already digested sample by electrophoresis method. I will explain how it is separated. Separate. The third step, separate this digested sample, digested with help of restriction enzymes, combination of restriction enzymes. After gives the breaks, now the separate the digested sample. Which method? By electrophoresis method. Next, transfer DNA fragments to probes. Transfer fragments to membrane. Transfer transfer fragments to membrane. With transferring DNA fragments to membrane, which is made up of nylon nitrocellulose membrane. After that, next visualization of specific DNA fragment using labeled probes. Next, after that, now transfer into the nylon nitrocellulose paper. After that, add radioactive labeled DNA probes. After separated this DNA for identification, use radio labeling probe. Now the final analysis the result. Analysis what is given final one. So hybridize the with radioactive label to detect by autoradiography. Can also non-radioactive latent system also. First, last analysis of result. Now let's see the result of the RFLP. And in this, first, before advantage, disadvantage, what they given. Right. DNA sub isolation. Let's write here. Isolation DNA. Is isolated DNA by restriction endonuclease cut. Cutting isolated DNA by restriction endonuclease. After with help of the restriction endonuclease, it gives the fragments. That means markers. Now these markers separate by gel electrophoresis. I will explain gel electrophoresis process by gel electrophoresis. Next, after separate these separated DNA transferred, separated DNA transferred out of gel onto a membrane filter. Membrane filter. Already told you this membrane filter made up of nylon nitrocellulose. Nylon or nitrocellulose. Now, next step, radioactive labeling. Radioactive labeling. After radioactive labeling and southern hybridization, I will explain this one also. Southern hybridization. Southern, these are the steps. Southern hybridization. Southern hybridization. Now, the analysis RFLP pattern with positive bands. The final after southern blotting. Now I identify RFLP pattern with positive bands. So this is the basic steps in this RFLP. Now here are the two important things. What is importance of gel electrophoresis? How it is involved in this? First of all, gel electrophoresis. Well, let's take another slide. So first, write all this isolation DNA because I'm going to erase this. Isolate DNA with help of restriction endonuclease. Cut the DNA. The isolated DNA separated by electrophoresis gel and DNA transfer into the membrane filters, nylon, nitrocellulose. Later, adding radioactive probes, southern blotting, southern hybridization. So these are the important steps in RFLP. But in this, what are the mechanism in agar gel activities? Let's see. Uh, right here. Agrogel electrophoresis. Agrogel electrophoresis is the method of gel electrophoresis used in biochemistry, molecular biology, genetics, and clinical chemistries. In this, actually, the separate mixed population macromolecules such as DNA and RNA by agarose. In this, so agarose gel electrophoresis. In agarose gel electrophoresis, it is the mainly separation of DNA or RNA by agarose gel. First, what is agarose here? Agarose is a liner, the polysaccharide. It is a polysaccharide made up of basic repeat unit of the agarose. It is one of the components of agar, that is mixture of polysaccharide from seaweeds. It is extracted from the seaweed algae. It is it is used at concentration between 1% to 3%. Agrogel is formed by suspending dry agarose in aqueous buffer and then boiling mixture till it becomes clear solution, which is then poured and allowed to cool room temperature form the rigid gel. Liquid to transfer into the solid. Then only it is utilized. The gelling properties attributed inner and intramolecular H bonding between the long agarose chain. So, in what is the mechanism in gel for electrophoresis? In gel electrophoresis, separate DNA fragments by size. Actually, this is agarose gel electrophoresis technique. After that, we let's take color. Already we have taken separated DNA and utilized restriction endonuclease. We have the uh, different length of DNAs that we have taken and the agarose gel, different type of fragments agarose gel. Now these samples pipped out on the agarose walls. Later what happened? If the sample DNA are pipetted into the sample wells, the followed by the application of electric current. Now the electric current applied to gel. The negative and which cause the negative charged DNA. Actually, DNA is the negative charged DNA. The migrated into the electrophores toward the positive ends. From this, after applying the electricity, this different length of DNA is filtered through this agar gel, and later the separated based on the length. For example, this small KB here and large one here. So this medium one here. So this type of patterns formed in agar gel. So these DNA fragments take up the dyes as they migrate through the gel, and emulsion with the ultraviolet light, it causes the intracalyctated dye fluorescence. The larger fragment fluorescence more intensely, and the smaller fragments include less mass of DNA. So like that, based on the length, we have to separate the DNA fragments or markers. So anyway, these are the separated with help of gel electrophoresis method. After separation, another important method. Actually, after separation, using radioactive labeling. So this is the radioactive labeling probes. This is for detection of the particular nucleotide sequences. After that, after separation, the best another method is southern hybridization. In southern hybridization, in southern hybridization analysis, it is a method of detecting specific DNA sequence. Already you separated by electrogel and using probe. Finally, you detect the specific DNA sequence in a genomic DNA sample. Already you separated various DNA. In that involves the transfer of DNA fragments separated by electrophoresis onto a membrane, nitrocellulose nylon membrane, and subsequent fragment detected by the final one detected by hybridization using specific probe labeled with fluorescence dye. Already we adding the probes. This the identifying the separated DNA with help of the probe, that is called southern hybridization technique. That is called southern hybridization technique. So in the final, after steps, RFLP steps, advantages and disadvantages. Right. So don't forget the steps. This advantage RFLP, they codominant. Already told you what are the dominant or codominant just beginning. They are the type of codominant measures variations of a level of DNA sequence where, as it is in polymorphic, not protein sequences. Not this is for not protein sequences, not recognized for protein, only for DNA. The RFLP loci are very large, so even very small segment of chromosome can be mapped, and also study phylogenetic relationship. Locus, actually the phylogenetic relationship based on the locus. So this RFLP also the loci very large, so even that's why it is useful for the studying phylogenetic relationship. Next, very reliable marker and easily detecting the DNA molecules. So these are the advantages. And let's see the disadvantages. Requires relatively very large amount of DNA. Of course, it is a short, short fragment. But to take the fragment, first of all, you separated the large amount of DNA. That is the disadvantage. Requirement of radioactive probe makes the analysis expensive and hazardous. This radioactive very expensive and it is harmful to us also. And they are not useful for detecting single base change point mutation. For this, it is detecting not only single. For detecting, we can apply the more nitrogen bases. This is also one of the disadvantages. It is time consuming, laborious, and expensive. The level of polymorphism is low. The polymorphism the very low in this type of uh, markers, pol markers. Uh, any other potential? No need, no need. Okay, right. So next hybridization, non-PCR completed. Now PCR based technique. PCR based technique RAPD, ISSR, SSR, AFLP, EST and SCOT. RAPD. I will explain later. Randomly amplified. Another I forgot one thing. RFLP, another important gene mapping. Mainly it is used for gene mapping. And RAPD, random amplified polymorphic DNA. The randomly it is works as a two types. It is act as a restriction endonuclease and also PCR too. But in the restriction endonuclease, RFLP only restriction endonuclease enzyme act as a enzyme, but not PCR mechanism, but not shows the PCR mechanism. But random amplified polymorphic DNA shows. Here, randomly amplified polymorphic DNA, RAPD, are genetic marker resulting from PCR amplification. This is important. PCR amplification. PCR amplification of genomic sequence recognized by primers recognized by which primers? Lenmer random primers. Lenmer random primer attributed nucleotide sequence. Which is a dominant markers. RFLP codominant markers, but RAPD the type of dominant markers that requires no prior knowledge of DNA sequence like RFLP, which makes them very suitable for investigation of species that are not or well known. The first of all, the later steps first, right. First, this is a DNA template. This DNA template, after separate with restriction endonuclease enzyme to breaks. After fragments formation, it is continuation with PCR mechanism. That means these fragments the produces more and more copies. But RFLP only what the fragments get, then they are only utilized for the further mechanism. But here after getting fragments, these fragments makes multiple by PCR mechanism. After the rest of the same, gel electrophoresis amplified and later finally southern hybridization separated. So these are the common and the simple principle in this RAPD. The RAPD analyzes PCR based molecular marker technique already told you. Polymorphism is detected by using oligonucleotide here. First of all, detect oligonucleotide 10 bases long random sequence of primer reaction in strain which has in its genomic DNA sequences complementary to the primer oligonucleotide. I will explain later. Here, basic steps what they've given here. Okay. First of all, this is DNA. This is DNA template. From the DNA template, isolation. Isolation. After isolation, this DNA fragment from the DNA. So actual, sorry, genome. From genome, you separated the DNA. The DNA act as a template. This template adding talk DNA polymerase, sorry, talk DNA polymerase. Already told you the function of talk DNA polymerase. Primer, TNTP, and buffer. These are adding to the DNA template. They are adding to the DNA template. What happened? And kept in thermocycler. What happened when you add this? Automatically, as it shows the PCR mechanism also. What are the principal steps of PCR mechanism? First of all, the DNA template, denature, denature separated, denature. After denature, annealing. What happened during annealing? During annealing process, here the primers, what are there? There these primers attached at 3-hydroxy end primer. For example, this is one strand, this is another strand, 5 and 2, 3 and 3 and 2, 5 and. So always the primers attached in annealing process, primers attached at here. And last extension. During extension, Taq polymerase, DNA Taq polymerase also adding towards 3 end. Now it is extended towards the 5 end direction, like this. Right? Now complementary strands are formed. After the complementary strands are formed, now these are amplified products separated into gel electrophoresis. What happening gel electrophoresis based on the length and molecular weight they are separated. Molecular length and weight they are separated by using southern hybridization. Southern hybridization by using probes, radioactive probes. That's it. And let's see the advantages and the best advantage of this RAPD DNA fingerprinting. Mostly it is useful for DNA fingerprinting, and it need for small amount of DNA, 15 to 25 nitrogen bases are there. Makes it possible to work with the population which are inaccessible for RFLP. Already one of the disadvantages RFLP, it is not accessible for all condition. Those there not accessible, in that situation we can apply the RAPD. It involves non-radioactive SEs. Sorry, actually I told after gel electrophoresis we apply the radioactives, but we apply the radioactive in RFLP. But in this in RAPD need not to apply any radioactives, any radioactive probes. Why? Because already the RAPD itself act as a probe, because with the help of the probes, it is recognized the sequence of the DNA. That's why no need to apply the non-radioactives. No need to apply the radioactives. It needs simple experimental and setup requiring only thermocycler and an agrogel assembly. Just after adding template, Taq polymerase, buffer, primer, and placed in thermocycler. That's it. After that, apply to the agrogel, gel electrophoresis. But what is the extra in RFLP? In RFLP, we use radioactive labels. Already told you disadvantage the radioactive label. The radioactive label utilizing most expensive and hazardous to us, harmful to us. It causes cancer. It causes cancer. That's why it is not preferable. And this RAPD not require species-specific probe library. This work can be conducted in large variety of species where such probe library are not variable, are not available. Sorry, are not available. So no need to require here the species-specific probe. But in RFLV, we separated the one specific probe, but there is no specific probe. Work can be conducted large variety of species, not only within the one species. In RFLP, taking one or two species, but here we can take the large variety of species where such probes library are not available. It provides quick and efficient screening for DNA sequence based on polymorphism at many loci. It does not involve blotting and hybridization steps. But RFLP blotting hybridization step involved. But here, no autoradiography, no southern hybridization. And RAPD markers have been successfully used for the following applications such as construction genetic mapping, mapping of traits, analysis of genetic structure of population, this is important, fingerprinting of individuals, targeting markers to specific region of the genome, identification of somatic hybrids, a result system of evolution, phylogenetic evolution also utilized for this RAPD markers. First of all, construction of gene maps. Their potential as genetic markers for map construction has been exploited, and map consisting largely or exclusively of RAPD markers have been developed in Arabidopsis pine Elianthraxa. And some of the here, apart from the advantage, some of the limitations also there. The RAPD polymorphism are they inherited as dominated and recessive character. This causes a loss of information relative to markers which shows codominance as it inherited and as dominated recessive character. Dominant recessive character, the loss of relative markers which shows codominance. So as it already shows the dominant character, there is here the losing information about the codominance. And another limitation, RAPD primers are relatively very short mismatch. Even the single nucleotide can even prevent the primer from the annealing. Oh, and there is loss of bond. As the RAPD primer very loss, if actually this primer is complementary to the template stand, but any one nucleotide mismatch, it not act as the complementary. As it not act complementary, it not produces annealing process and not extension process. The production of non-parental bonds of offspring known as pedigrees warrants its use the causation and extreme care. And it is very sensitive change in PCR condition resulting in change to some of the amplified fragment. The variations of RAPD techniques are the DNA amplification fingerprinting and the attribute polymerase chain reactions. So these are the important applications of RAPD. And they are completed. SSR. Another polymerase chain reaction based markers, SSR, simple sequence repeat. Simple sequence repeat the given right. SSR, simple sequence repeat, or micro satellite. Another name micro satellites. The first of all, uh, simple sequence, macro satellite, simple sequence repeat, short tandem repeat STR, a repeating sequence 2 to 6 base pair. DNA, it is a type of variable number of tandem repeat. Microcell typically codominant. This is codominant sequences. So the term microsatellite was first coined by Litt and Lutty. Right. So that the tandem arrangement repeats mono, di, tri, tetra, penta nucleotide with a different length of repeated motifs such as, for example, this repeated, like here given already clear. AC, 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 AC. It is 2, 2. TG, 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 TG. So this is repeated. So these repeats are widely distributed throughout the plant and animal genome that display a high level of genetic variation based on differences in the number of tandemly repeating. This repetition can called as tandemly repeat. Tandem repeat. The macrosatellite repeats were earlier called variable number of tandem repeats. It was described as a location in a genome where short nucleotide sequence is organized as tandem repeat. The where in genome where the tandem repeats are present, that region separated as a marker. This can be found on many chromosomes and often shows variation in length between individuals. So this repeatedness, the not a same, not a unique. It is dynamic. Some variation shows in this between the individuals. Each variance act as inheritable allele, allowing them to be used personal parental identification. This tandem repeat, this tandem repeat actually inherited alleles that mean from pass through ancestor to the next offspring. That's why this is analysis useful for genetics and biology research, forensic and DNA fingerprinting process. Repeated DNA representing over the 40% of human genome is arranged in a different array pattern. The repeats were first identified by the extraction of satellite DNA, which is present at the short chromosome part, which does not reveal how they are organized. The use of restriction enzyme showed that some repeat blocks were interspersed throughout the genome. DNA sequence later showed other repeat are clustered at specific location, with the tandem repeats being more common than inverted plants, so inverted repeats, which may interfere DNA replication. So this repetition interrupt the DNA replication process. Uh, Based on number of nucleotide repeated, here the mononucleotide means only one repeated in this. Maybe only one nucleotide, you are given A, adenine. Actually, adenine, thymine, guanine, cytosine, guanine. Like this, after formation like this, repeated start. Let's change color. Single nucleotide, adenine. Next of the common. So this repeated sequence only, and let it be this sequence, only write the repeated sequence. And next dinucleotide sequence. Dinucleotide sequence, dinucleotide sequence, CA, 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 like that. Trinucleated, common CGT, repeated CGT, repeated CGT, this common. Tetra, CAGA, again repeated CAGA, again repeated CAGA. Penta, AAATT, AAATT. So like that. The number of repeated forces based on the arrangement of nucleotides in the repeat motif. Uh, they are classified based on the nucleotide number. So here SSR polymorphism, common CTT, 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 trinucleotide repeat, gel electrophoresis, P1, P2. So like this patterns formed, if you see in gel process. So this repeatedness also based on the molecular weight and length separated with help of gel electrophoresis process. Advantages of these markers, it is tend to be highly polymorphic. When compared with RFLP and even RAPD, which is highly polymorphic. The genotypic throughout the the genotypic is high. This is simple PCR assay like RAPD. Many SSR markers are multi-allelic and highly polymorphic. Most SSR codominant and locus specific. No special equipment is needed for performing SSR assay. However, the special equipment is needed for some assay methods. Startup costs are low for manual assay methods. SSR assay can be performed using very small DNA samples. So SSR markers are easily shared between the laboratories as it is taught in this. So the development SSR labor intensive, costs and are very high cost. Then let's see the application. Genetic diversity analysis, because this repeatedness of sequences, individuals, not only individuals in different species, also different. Based on the repeatedness, we can find out the genetic diversity and cultivar identification, phylogenetic relationship. Already told you, phylogenetic relationship, gene mapping and identification of qualitative traits, marker assisted selection, MAS, sex determination of DSS plants, EST-SSR, and expressed sequence tag, EST, short subsequence of cDNAs. It is the sequence of cDNA. From DNA, repeated sequence, that is SSR. But EST, an expressed sequence tag, it is short subsequence of cDNA. Expressed sequence tag obtained from partial random sequence of cDNA library. How the cDNA library complementary DNA, the cDNA formed by reverse transcriptase. That means First DNA to RNA, again RNA to DNA, that is cDNA molecules. It is made up of 300 to 500 nucleotide long mRNA sequence from many genes expressed in sample organism. S are present in EST region known as ESS or SS or genetic microsatellite. So exon 1, exon 2. Then what happened here? In genomic library, first genomic DNA, exon. The light colors are introns. So later what happened? These introns are removed. In cDNA, no introns, no introns. That's why it is effective. Next, inter simple sequence repeat. Inter simple sequence repeats. Any other about SSR directed PCR? In your book, SSR, I think ISS are not given. Selective amplification microsatellite SSR only attributed to an APPCR simple sequence SSR micro only. DNA application fingerprinting not given. Not given. No need to explain this. Enter simple sequence repeats. No need. Scott also not given. And AFLP, amplified fragment length polymorphism. This is combination of PCR and RFLP. PCR and RFLP, both informative fingerprinting amplified fragments. AFLP technology is a DNA fingerprinting technique that combines RFLP and PCR, already told you. It is based on the selective amplification subject of genomic restriction fragment using PCR. So let's see the AFLP process. First step, common is digest the genomic DNA with restriction enzyme. This restriction enzyme digested with help. Next, ligate commercial adapters, defense sequence. After cleave, after separated DNA, again ligate attached with help of ligate adapters. Now, now carry out PCR and the adapter ligate mixture using primers that target the adapter, but they vary in the base at the three end of the primer. Oh, that's it. Single nucleotide polymer. So I will explain later, but let it complete AFLP into two steps. Actually, this AFLP highly sensitive method for detecting polymorphism throughout the genome, and it is becoming increasingly popular. It is essential a combination of RFLP and RAPD methods. Already told you, and it is applicable universally and is highly reproducible. That means production is more. It is because of it already it shows the PCR mechanism, polymer chain mechanism. It gives the multiple copy. It is based on amplification of genomic restriction fragment generated by specific restriction enzymes and oligonucleotide adapters. Of few oligonucleotides, these oligonucleotide adapters actually act as a primers. It is novel DNA fingerprinting technique. The DNA fingerprinting involves the display of set of DNA fragments from specific DNA sample. Fingerprintings are produced without prior sequence knowledge using a limited set of genetic primers. AFLP technique uses straight net reaction conditions for the primers enabling and combines the reliability RFLP technique with the power of PCR technique. First of all, here, uh, actually not given the steps. First, actually, we have taken DNA strands. With help of restriction enzyme, DNA strands cut into DNA strand with restriction enzyme, generally two enzymes. The double stranded. Oligonucleotide adapters are ligated to the ends of the DNA fragments. After actually it's cut as a, for example, so DNA sequence 5 and GAATTC TTAA 3 and CTTAAG AATT. Now with help of restriction enzyme, it is cut as a like this. Let's change color. AATC like this. Cut in different direction. Right. After that, it produced AATTC T or here GAATT. Now this stands with adapter ligation utilized. That means here AATTC. It is ligated by TTAA, joined by TTAA, like this. And here AT. AT means here ligated by TA. This place ligated by TA and this place ligated by TTA. So anyway, in the second step, adapter ligation occurs. After adapter ligation, later PCR, polymerase chain reaction. It shows PCR polymerase chain reaction. In PCR polymerase chain reaction, this is formation into number of copies. This is one strand. For example, this is one strand, this is second strand. This one strand, one cycle would produce two strands. Second cycle, four strands, 8 strands. It is multiple in number. Like this number of DNA strands are formed. So after the selective amplification, a set of restriction fragment usually carried out with help of the P32 labeled primers designed according to the sequence of adapter plus 1 to 3 additional nucleotide. Only fragments containing nucleotides, these fragments containing the restriction site sequence plus additional nucleotide will be amplified. That is like a PCR mechanism, annealing, denature, annealing extension. The third, gel analysis of the amplified fragment. After amplification product, separated and highly resolving sequence by gel electrophoresis, visualizing using autoradiography and fluorescent and silver staining techniques can be used to visually products. So the restriction fragment for amplification are generated by two restriction enzymes, rare cutter and frequent cutter. Rare, that means that is some rare name itself given rare cutter, and one other one is frequent cutter. Uh, uh, that's it. IFLP adapter consists core sequence enzyme, no need. The restriction fragment, no need. Right. So this is the steps in AFLP. Next. Advantages, and actually not given here advantages, but you remember these advantages of this technique. This technique extremely sensitive, and it has highly reproducibility, rendering a supporter to RAPD. It has wide scale applicability, providing extremely proficient, and it is not only simple fingerprinting technique, but can also be using for mapping, both the fingerprinting technique and also mapping, gene mapping. Are some disadvantages. It is highly expensive, requires more DNA than in needed RAPD, more DNA also more, and it is technically more demanding than RAPD, that is require experiences of sequencing gel. AFLP are expensive to generate silver staining, fluorescent dye, and radioactive detect bonds. These are all utilized. The silver staining more expensive.